Okay, so I definitely recommend going back to this preview video and checking out how we did the mini prep. The mini prep was the procedure that we did first to isolate the DNA, but we'll do it again here really quickly. Okay, so buffer P1, remember we had just that pellet of raw DNA. That's just to resuspend those cells because those are all in a little cell clump. P2, this was a lysis buffer. Remember this turned this blue. This is lysis, so this breaks the cells apart. So now the cells, so they started up here, it's like a little clump. They're resuspended, lysis breaks them up. P3, this is called a neutralization, neutralize. So essentially, that just stops the lysis. That puts just neutralization buffer P3, that stops the lysis at 5 minutes. Now we had this big mess, remember, in our tube. And it was kind of like that, like all that white crap in there. So we did a spin, and then when we had on our spin, we did a big spin, we had all that junk, remember all that like fat from the cells and all that plasma membrane, but then we had a supernatant. And this is where our DNA was, right here. Okay, so then remember we went to a new column, and this is the special one, this is not a regular tube. This was the spin column. Remember it had a little filter right there, and then it was in another tube, so all this bad stuff can go in. So essentially we took this supernatant, which contained our DNA, we put it in here, and we spun it one more time. So we did a big spin, that's a spin. What we ended up with was we got all the junk in the bottom again that we spun down because the centrifuge pushes all that down, and the DNA actually bound to the filter. So remember that filter I hopefully showed you, that's what this spin column does. It actually catches the DNA in that little like white little filter. Go in your book and take a look at this again. So let's restart this at that point. So now we're in the spin column still. Remember this filter right here, it has caught all the DNA and it's just sitting there. And remember we have this little tube right here catching everything else. So all that stuff that we got from the first spin, it's down here. Then we did a wash. So this is just a chemical wash. This washed some more stuff down, cleaned the DNA. Then came the final step. So our DNA is still stuck up here until we add what's called the elution buffer. So the word elution means, and we're just gonna call elution buffer EB. Elution means to unattach something, means to elute it, means to get it down. So essentially, if this is our spin column, here, let's do it over here. And we put our EB in. So remember the wash just went down here and we emptied everything out again. So for EB, remember we actually got a new tube, like a nice new one that was gonna go under it. Because when we added the EB, it got to the DNA and it unattached it from the column. And when we spun it, our DNA is now in this EB. So our DNA is still in there. EB was the thing that disattached our DNA from that tube filter, from the column spin. And then, there we go, we had our DNA. Oops, perfect. And so the DNA we were looking for, remember, was that big plasmid. And remember, plasmids are circular DNA. Bacteria can exchange them, we can use them as tools to express genes inside of other organisms. So what we did next was we wanted to make sure that this was actually what it was. So we had two enzymes here. I'm gonna redraw everything. So this vector, or this plasmid, you can do this, call them the same thing. It was like X amount of base pairs. Yeah, it was like, you'll have this on any test questions for this. Like the numbers and everything will be all there. But essentially you had that PVU2, and you had, oh, and remember, this is the thing that cuts DNA right at that like open spot, remember? So this is what you end up with when you cut with PVU2. Just a very specific area of the DNA that used to be here. PVU is that restriction enzyme that can cut right here, and you have these sticky ends, they can re-anneal to something. So that's important for later uses. We ended up just cutting and just visualizing that. That's fine, and we had two more over here, did the same thing. They think they were AVA1. So be able to like measure where the distances are in these little segments because that's important. And we did that next. essentially we had, let's make sure here, we had a gel, and DNA is inherently negative. So DNA wants to go this way, wants to go towards the positive. So let's say our DNA, but remember the first thing we had was the ladder, and ladder is just completely, we just buy that, and that's essentially just a size. Like the bigger ones stay on top, and the little ones, they can speed through the gel. So here's our big gel right here. So remember the ladder, that's just a size thing. So like, let's say that these are like, 10, 5,000, 2,000, 1,000, 0.5 or something. Ladder's just for size reference. When we loaded up our cut ones, so one cut, remember, so if we cut right there, we would have like 2 segments, because this is the segment that got cut out. Another cut, but from another enzyme, be a little bigger, smaller. 2 cuts was 2 larger segments, oh, sorry, be down here. 2 larger segments and then 2 really small segments, still are these 2 are right here. Sorry, this is getting a little weird. You'll see this on the, you won't have to like draw any of this or anything. Basically, DNA is attracted down the gel towards the positive charge, away from the negative charge, and the larger segments are up here. See where the ladder will show you that, the larger, bigger segments, they're too big to get through the agarose sugar gel. But the smaller segments, they can just speed right through. So that's how you can measure those. So essentially what we did in this lab was we isolated DNA from bacteria, we cut it with restriction enzymes, and then we visualized it on this gel. Oh yeah, and we can do like 0 cuts here. So the 0, 1 will just be like one big thing, and then maybe like one little one. That's just if it like sped through a little bit more, because it's circular. But that is essentially what we did in that lab. Isolated DNA, cut it, visualized it.